let's start with the bones. As we know, bone has two, and histologically we divided in two parts. The inner spongy bones, as you see in these slides, contain the bone marrow, and outer bone is a compact bone, is contain calcium, calcium phosphate, and really hard tissue of our bodies. Okay, and okay, I need to now the a little bit about the histology of compact bone. The hard part of our bones, as we see, okay, the osteon, the compact bone consists of units we call osteons, are Haversian systems. They containing, as we see here, is a unit of Haversian systems, and it contain osteocytes which trapped in lacuna pits, as you see here, in a concentric circle. And Haversian canals, they contain blood vessels, as we see in these slides, and nerves which run through osteons. Okay, osteon as a unit of the compact bone consists of a Haversian systems. As we see here, there are osteocytes trapped in a lacuna, which is pits, and also we have bloods and nerves in Haversian canals in our compact bones. Okay, lamel which are concentric layers. Here, as we see, the concentric layers of matrix containing calcium, mostly calcium phosphate, a hard tissue like a stone. Okay, here this slide is really important. You have to be familiar with the part of the bones, long bones, okay, which kind of part we have in the long bones, as we see here in the one long bones. An example, we have epiphysis, okay, two epiphyses in a one bones, rounded the end of the long bones, which should be here, okay, and contain red marrow, okay, and the epiphyseal plate is very important where bone growth occurs, okay. Epiphyses are really important part of our bones because they contain marrow, okay, which the bone cells are growing and multiplying, okay, and also is important epiphyse because it is a place where bone growth occurs. Okay, the other part of our long bones is articular cartilage. As you see at the end of both sides, we have articular cartilage, which cover the epiphyses to prevent bone from rubbing on bone. It is also important. And we have diaphyses. The midsection of the long bone contain yellow marrow, as we see here. And we have Uh, endosteum, okay, is the thin layer of a connective tissue that line the surface of the bone tissue to form the medullary cavity of long bones. Here we see the medullary cavity and surrounded by endosteum. Lastly, the periosteum is a membrane that covers the outer surface of our bones. These are the parts of our long bones, okay? Epiphyse is really important here because it has two main functions, okay? It contains red marrow, which the stem cells are included, and then they produce cell for us, and also it is a place, a location where bone growth occurs. Okay. Now let's talk about the bone cells. We have a really important 4 type of bone cells, and each bone, these kind of cells has a important function. You also need to know the bone cells and their function. As we see here, osteoblasts, the immature cells that form orbital bone, okay? The cells that form bone are osteoblasts. They secrete the bone matrix made up of calcium phosphate and collagen fibers. As the matrix form around the osteoblasts, these trapped osteoblasts, they become osteocytes. Okay, we also have here osteocytes. The osteocytes are a kind of osteoblast which trapped by the matrix, bone matrix. Okay, and we have also osteocytes as here. Okay, which have osteoblasts are immature cells which form bone matrix. Okay, once bone matrix builded, then this will surround these osteoblasts, and these osteoblasts become a mature cells which we call osteocytes. Osteocytes are the mature form of the osteoblast. And osteoclasts, other important class of bone cells, okay? As you see here, these cells are multinucleated. There are, as you see, several nucleus in the cells, okay? Osteoblast is has only one nucleus, as you see. And these cells are important because they resorb or break down bone by secreting enzymes that break down collagen and calcium phosphate crystals. Okay, as you see, osteoblasts, they form bone. Osteoclasts, they resorb, they break down the bone which is built by osteoblasts. Okay, osteoblasts and osteoclasts work in balance to maintain the shape and model bones for growth and development. This is really important for our health, to maintain the health of our bones. Okay, the balance between osteoblasts and osteoclasts are really important because osteoblasts they build bone, collagen matrix, the matrix of bones. Osteoclasts will remove these collagen matrix. Okay, the balance between two cells are really important. Okay, as we know, bones are modeled during growth and remodeled continuously as stress and strain are placed on the bones. Okay, in during our lives, the balance of these two cells are really important. Okay, as we child age, the osteoblast should work much than the osteoclast in order to grow uh, um, our bones. Okay, but once we are adult, in adults should be these two cells work in a balance. Okay, in some case, in continuously as a stresses or strain, which will affect the balance between these two cells, and osteoclast will work too much as the osteoblast, and we lose the matrix of our bones, which is make our bones sensible to the fractures. Okay, as you see here, okay, here, once osteoblasts are working harder than osteoclasts, bone will grow. Okay, because osteoblasts they build bones. Okay, however, when osteoclasts they work harder than osteoblasts, then we lose the bone. Okay, the balance between two type of cells is really important. Okay, there are some diseases that we lose our bones, especially the collagen, the matrix of our bones. It is really important for our life to maintain our health. Okay, as we see as an example of imbalance between these two cells, which we call osteoporosis, you may know from the especially as we see commonly in the elderly women. Okay, osteoporosis can be seen in degenerative bone disease. Okay, bone resorption by osteoclast, because osteoclast's main function is resorption, takes place more rapidly than bone formation by the osteoclast. Then patient lost bone mass and exhibit fragile bones that are susceptible to fracture. Okay, if you lost the collagen form of the bone, then your bone will be a fragile bone, and these bones are susceptible to fractures. As you see here, the shape of the spinal cord. Okay, here we have a normal spinal cord. Once osteoporosis occurs, then as you see, the shape is changed. Also, the structure of bone. Okay, in the osteoporosis bone, a bone look like a spongy. Okay, spongy bones increases, and this type of bones are fragile bones and susceptible to fractures. Okay, here now, as an example for the previous lectures, the cells form bone matrix. Which cells form bone matrix? Anybody can give an answer? There are two types cells in our body important. One kind of the cells which form bone matrix. Okay. Who is brave to give an answer? Good. Okay. As you know, osteoblasts, okay, are the cells form bone matrix. Osteoclasts are the bone cells. They resorb the bone matrix. Okay. Osteocytes are the mature form of the osteoblast. Okay, good. And here the second question, the part of long bones at which bone growth occurs. Okay, these are the part of the long bones, endosteum, articular cartilage, epiphysis, diaphysis, and medullary cavity. Okay, at which part of the bone growth occurs? Okay, can I see your answer? Okay, here. Okay. Any answer about that? Okay, the part of long bones at which bone growth occurs. Okay, okay. C, yeah, correct. Okay, now, okay, epiphyses are important because of two reasons. Okay, once epiphyses are the place of the red marrow, okay, the place which stem cells are located and they produce cells for us, and also important because these epiphyses are the place which where bone growth occurs. Okay, good. Now let's go forward and then see a new stuff. Okay. Oh, we have here another question. Okay, if any balance between osteoblast and osteoclast is broken in favor of osteoclast, result in a disease known as osteopenia, hyperostosis, osteomyelitis, osteoporosis, osteomalacia. Okay, let's see your answer. Correct. Okay, D is the answer. Osteoporosis is a imbalance between osteoblast and osteoclast in favor of osteoclasts. Okay, if osteoclasts are hyperactive than osteoblasts, then we lose the matrix of bone, and this bone will be susceptible to fracture, which we call osteoporosis. Good. Okay, now, okay, let's talk about the skeletal muscle structure. Okay, in the rest of the time, we are going to discuss the structure and function of our muscles. Okay, they are associated to each other. If you know the structure of the muscle, and it really will be helpful to understand how our muscle working. Okay, the mechanism of action for muscle contraction is really important. Okay, muscles such as biceps, as you see here in the arm, consists of many fascicles or a bundle of muscle fibers. Okay, muscles consist of muscle fibers. Okay, and muscle fibers, as you see here, are the elongated cells of the skeletal muscles. Inside muscle fiber, there is another subcellular level of organization. I will talk about that, but muscles are a bundle of muscle fibers. Okay, okay, and then muscle fibers consist of other cells, we will talk about them. As we see here, okay, skeletal muscles consist of a long striated cells, which we call muscle fibers or myofibril, okay, with many nuclei organized in a bundle and wrapped by connective tissues, okay. The bundle of the myofibril, okay, and there are striated cells, as you see the appearance of the skeletal muscles, okay, and sarcolemma, as you see here, okay, the plasma membrane of the muscle cells we call sarcolemma, and this plasma membrane that has multiple inward extensions to form, as you see, T-tubule, okay, and we also have sarcoplasm. Sarcoplasm is the cytoplasm of the muscle fibers, okay. Sarcolemma, okay, the plasma membrane of the myofibril. Sarcoplasma is the cytoplasm of muscle fiber. Okay, let's divide, okay, dive into the muscle structure a little bit more. Sarcoplasmic reticulum, okay. I am sure that you know endoplasmic reticulum.
I just studied the slides.
Okay. Sarcoplasmic reticulum is the endoplasmic reticulum of a muscle fiber. Okay, as we see here, and myofibril, which are made of the two myofilaments. Okay, and these are thick filament we call myosin filaments, consists of a protein called myosin, and thin filament, which is actin filaments, consists of mainly of the protein as which is known actin. Okay, what does means? Okay, myofibrils are made of two filaments, myofilaments. The thick one is myosin filaments. The thin one is actin filaments. Okay, here a big picture of the same slice. We have myofibril, which is a bundle of the muscle cells, sarcoplasmic reticulum as the endoplasmic reticulum of myofibril, sarcolemma, the plasma membrane of the myofibril, and we have myofilaments, which are actin, thin filament, and myosin, thick filaments. Okay, sarcomere, okay, are the basic unit of muscle contraction containing myosin and actin filaments. What does means? And the two components of the contraction of the muscles are actin and myosin filaments. Okay, sarcomere is a basic unit of muscles contraction. Okay, as you see here in the slides. Okay, the sarcomere will begin with Z line and end up with the Z line. Okay, between two Z lines, we have one sarcomere, one unit of the muscle, which we call sarcomere. But the muscle consists of many sarcomeres. Okay, sarcomeres is a basic unit of muscle contraction and begin with, it is between two Z lines, okay? As you see here, the Z lines, and here also the Z lines. I will go forward and discuss a little bit in details, okay? Sarcomeres are joined at their ends of by Z lines, okay? Now, this is a, as you see, ultrastructure of our muscles, okay? A unit which we call sarcomere. As you see here in these slides, okay, myosin and actin filaments are overlap lengthwise in muscle fibers and producing a pattern of striation in the striated muscles, which is I bands, as we see here, A bands, and H bands. Okay, now let's see what consists of the I bands, A bands, and H bands. As you see in this circle, I made a red circle here. I bands is actin filaments only, okay? Actin filament of two adjacent sarcomere made I bands, and A bands is made of actin and both myosins, okay? Myosin and actin filaments both together, they make as an appearance of the muscles A bands, and H zone, as you see here, here H zone is only included myosin filaments, okay, which is a blue circle, okay. H bonds only myosin. A bonds, as you see here, A bonds both myosin and actin. I bands only actin, okay. As I said, the overlapping actin and myosin filaments produce the striation of striated skeletal muscles, okay? This is the reason why striated muscles look like striates, okay? Here, this is a big picture of the previous slides. We have here only myosin, okay, and in the H zone, in the H zone we have only myosin filament. In the A zone we have both myosin and actin filament, but I band only include actin filament, the thin filaments. Okay, now the question is that how the muscles are contract. What happens? Well, okay, what should occur to initiate muscle contraction, and what should occur to end the contraction, to get relaxed of the muscles? The mechanism of action of muscle contraction is really important. You will face many questions about the mechanism of action of muscle contraction. Okay, the most common accepted mechanism for that we call sliding filament model. Okay, sliding filament model of muscle contraction is really important to you. Now I am going to tell step by step, and then we will repeat it in rest of the slides, okay? A muscle contraction occurs when sarcomere shortened, okay? As you see here, this is a normal relaxed sarcomere. Okay, when muscle get contract, and this sarcomere will shorten, as you see in the last part, in the bottom of the these pictures. Okay, actin and myosin filaments slides pass on another and increasing their overlap. Okay, keep in mind actin and myosin filaments do not shorten. They slide on pass on and pass one another. Okay, actin and myosin filaments slide past one another. Okay, they don't shorten, but the sarcomere unit shortened. Okay, and a result of this mechanism, I band and H band decrease in length because actin and myosin filaments slide past one another, and therefore I band, which we know include actin filament, H band, which is made of myosin filaments. Once they slide past one another, that means I band and H band will the length of these bands will decrease. Okay, now the question is that here sarcomere contain which of following proteins? Now is your turn. Okay. Okay, good. Let's see your answer. Good. Good job. Okay. Myosin and actin are a filament, myofilament, or sarcomere units. Okay. Sarcomere is a basic unit of muscle contraction, and it consists of two proteins, two filaments, which are myosin and actin. Okay. Skeletal muscle produce movement by pulling on tendon, which are connective tissue. You know, tendon and ligaments. Tendon attach muscle to bone. Okay, that attach to muscle to bone. Okay, and skeletal muscle produce movement by pulling on tendons. Keep in mind, okay, muscles only can pull the tendons, okay, because they only can contract, okay, and by contraction they shortened, they get short, and then they pull tendons and result in movement of our bones in our body. Okay, and skeletal muscle acts antagonistically to one another. An example here, okay, once biceps make contraction, and triceps should make extension. Okay, the extensors serve the opposite purpose. Okay, otherwise, what would happen when two of them contract at the same time? Yeah, I mean, you can't get any movement, any motion. Okay, therefore the antagonists, they should work antagonistically. Okay. Okay. Now, step in muscle contraction is really important. You need to be familiar the mechanism of action of muscle contraction step by step, how we initiate muscle contraction, and how its the process is going forward and end up with relaxation. Here at the junction, okay, at the junction of the nerve and muscle, as we know from our previous lectures, acetylcholine vesicles are released. There are acetylcholine at the junction of the nerve and muscle, okay? You may remember effector organs, okay? The junction between muscle and nerve, there are acetylcholine vesicles. They release once action potential reach to the junction through the axonal neurons, and then it results in release of acetylcholine from vesicles. Okay, acetylcholine binds to the receptors of the sarcolemma, which is the membrane of the myofibrils, and causing sodium influx into the muscle fiber. If you remember the action potential of the nerve, it is similar action potential is happening in our muscle cells. Okay, once sodium influx into the muscle, as you know, the negative charge, relatively negative charge inside of the cells will change and become less negative charge or positive charge, and then action potential will start it. The action potential travel through the T tubules, allowing calcium channels to open and release calcium into cytoplasm. Okay, for the contraction of the cells, calcium is really important. Okay, first the contraction will begin with acetylcholine vesicles, releasing of acetylcholine from the vesicles, and then this acetylcholine will bind to the sarcolemma and cause influx of sodium into the myofibril, which result in releasing of the calcium from the sarcoplasmic reticulum. Okay, once calcium released from the sarcoplasmic reticulum, okay, this calcium will bind to troponin, a protein of muscle cells, which wrap around actin filament and exposes the myosin binding site on actin. And ATP is hydrolyzed to provide energy for flexing the myosin head. And flexing brings the actin filaments closer to the middle of the sarcomere, to the overall length of the sarcomere is shortened, and contraction is occurred. Okay, these are the steps of muscle contraction, step by step. Okay, first acetylcholine vesicles will release acetylcholine, and then sodium influx, and then calcium will bind to troponin, and then which allow the actin and myosin filament exposed to each other and slice on it and make the sarcomere shorten. Okay, I have a short video. If we have time, I will show you at the end of these lectures. Okay, now this is, I mean, a little bit as an explanation of these steps. Okay, first first step of muscle contraction is an action potential travel along motor neuron, as you know, toward to the skeletal muscle, and their point of connection is called neuromuscular junction, the junction between neuron and muscle. Once action potential reach this neuromuscular junction, and then it results in releasing of acetylcholine vesicles from its vesicles. This is the second step. In the third step, acetylcholine will bind to the receptors on the sarcolemma, causing sodium influx. In the third steps, okay, once you decrease the negative charge inside of the muscles, and the calcium channels will open, and calcium will release into the cytoplasm. As we see here, calcium ions bind to the troponin. Here we see in these pictures, okay, these calcium molecules, the green, okay, one we have here, the troponin, the tiny filaments, okay, and then calcium will bind to the troponin, and that is wrapped around the actin filaments and expose the myosin binding to the actins. Once the troponin is there, actin cannot expose to the myosin, okay? Once calcium bind to troponin and remove the troponin from the actins, it allow the expose the myosin binding to the actins, okay? And then we also need ATP as the energy sources, ATP hydrolyzed to provide energy for flexing by myosin heads. As you see here, myosin heads, once the ATP hydrolyzed, and then this myosin head will flex and then move the actin one step forward, okay? As you see here, okay, is bind to the actins, the myosin heads, okay, which also need energy from ATP, and then move actin one step forward. And then flexing past the actin filaments closer to the middle sarcomere, okay? Actin will slides, as you see here, okay? And then sarcomere is shorter, okay? If you contrast them, the sarcomere at the bottom of the slice is shorter than the sarcomere at the top of the slice, okay? In order for relaxation to occur, ATP must use the pump calcium back into the sarcoplasmic reticulum, okay? Once the calcium is attached to the troponin, the contraction is continuing, okay? In order to end the contraction and make the muscle relax, we need to get the calcium back into the sarcoplasmic reticulum, okay? Any causes which prevent calcium back into the sarcoplasm, and they result in muscle contraction, okay? To continue that, okay? Now we still have a few minutes. Then, okay, step in muscle relaxation. Okay, how we end up with muscle contraction? Okay, because we need muscle contraction if the body need it. Okay, but also we have to end this contraction and make muscle relax again, okay? For that, we need acetylcholinesterase as enzymes which break down acetylcholine. Recall that acetylcholine, acetylcholine initiate the muscle contraction, okay? Once acetylcholine released from the acetylcholine vesicles, okay, which result in sodium influx, and sodium influx result in calcium releasing into the cytoplasma, okay? But in order to end with contraction of muscle, we need to break down acetylcholine, okay? In order to stop the action potential, okay? For that, we have acetylcholinesterase in our body, which break down acetylcholine and end the process of muscle contraction, okay? Any drugs which inhibit acetylcholinesterase result in muscle contraction to continue, okay? This is really important. Some poisoning, they prevent acetylcholinesterase to break down acetylcholine, and then muscles will contract, all the body muscles, the muscle in which allow inspiration and respiration, and then we cannot inspire the air, okay? It is really important function for acetylcholinesterase, okay? Calcium ions are actively transported back into the sarcoplasmic reticulum once acetylcholinesterase break down acetylcholine, once the action potential is stopped, okay? Linkage between actin and myosin filaments breaks after calcium back into the sarcoplasmic reticulum, because calcium, one calcium in the cytoplasma is bind to the troponin and move the troponin from the actin filaments and result in actin to expose the myosin. When calcium back into the sarcoplasmic reticulum, that means the troponin will bind to the actin again and prevent actin myosin exposing. Okay, actin and myosin filaments slice apart, and then muscle fiber relax. Okay, contraction and relaxation of muscles. Okay, since two of these steps require energy, both contraction and relaxation of muscle require energy, a dead tissue cannot relax already contracted muscles, okay, which we call rigor mortis in the dead bodies, okay, because relaxation also need energy. Once the body, the cells die, and then they cannot make a relaxation because there is no more energy for relaxation, and result in contracted muscles, which we call rigor mortis. Okay.
Dr. It's 11:55.
Excuse me. Sorry.
Class over.
55.
Okay. Do you have a question?
No, we're saying class is over.
No. I remember it is. We have a few minutes, 5 minutes at least, because we started 11:05. Okay, but I mean this is my last slide. Okay, I will end up with this slide. Okay, energy for muscle contraction is important. We need ATP hydrolysis for both relaxation and contraction. Also, creatine phosphate is a storage form of energy in our muscle, is really important. Once ATP is over, and creatine phosphate will give its phosphate to the ATP, make ATP for contraction and relaxation. Glycogen is also a source of energy for muscle contraction, for midterm, and also fatty acids are long-term energy sources for our muscles. Okay. Okay, this is a last question for these lectures. Okay, regarding to sliding filament model of muscle contraction, which of the following statements is not true? Okay, myosin and actin filaments are organized into repeating units called sarcomere. This is correct. Okay, and I band and H zone decrease in length as a filament slides past one another. This is also true. As the sarcomere becomes shorter, the muscle fiber shortens. Is that correct? No, because muscle fibers, actin and filament, they don't shorten. Okay, they don't get shortened. Okay, only they slide on each other. Okay, but sarcomere shortened, I band and H band shortened, but filaments doesn't. Okay, releasing of calcium initiate the contraction, but at the beginning of the contraction, we need acetylcholine to release at the junction. Okay, don't forget these points. Okay, okay. Let's see you on Tuesday for a review session. Okay, if you have question, we will. I am ready to ask this question during the review session. Also, feel free to send me email about your question, about your concern, especially about the voice of the Zoom and the appearance of the slides. I need your feedbacks about that, and then if needed, I need to improve the slides or the speed of the internet. Okay. Okay, stay tuned with me, and let's see you next week on Tuesday at 10 a.m. Okay, and be ready for your exam, please. Okay, bye. Okay.
Doctor.
Okay, thank you. Thank you. I will end the meeting.